Hello, today we are going to talk about DNA and DNA replication. What is the double helix? The double helix is a term used to describe the structure or the shape of DNA. DNA looks like a ladder that has been twisted. Double means two, helix means coil. A few lessons ago we talked about macromolecules and we learned that nucleic acids were one of them. DNA and RNA are nucleic acids. DNA is used to store and transmit hereditary information, and RNA is used to make proteins. This is a picture of RNA, ribonucleic acid. RNA is single-stranded. This is a picture of DNA, deoxyribonucleic acid. DNA is double-stranded. The nitrogenous bases found in DNA are cytosine, guanine, adenine, and thymine. The nitrogenous bases found in RNA are cytosine, guanine, adenine, and uracil. The structure of DNA. DNA is composed of a long polymer of nucleotides, and nucleotides are composed of three parts. Every nucleotide contains a phosphate group, a 5-carbon pentose sugar, and a nitrogenous base. The backbone of the DNA molecule is composed of alternating phosphate and sugar molecules, and the bonds holding these phosphate and sugar molecules together are called covalent bonds, and they are very strong bonds. If we look on the inside of the DNA molecule where the nitrogenous bases are found, hydrogen bonds are holding these bases together. So covalent bonds are holding the backbone of the DNA molecule together. The covalent bonds are holding the phosphate and the sugar molecules together, and weak hydrogen bonds are holding the nitrogenous bases together. Replication is basically when DNA makes an exact copy of itself. Replication takes place in the nucleus. Any mistake in copying leads to a mutation. If the DNA has a mutation, then there will be a chromosomal mutation. If you think about it, a chromosome is nothing but DNA wrapped around proteins. So if there is a DNA mutation, then there is an chromosomal mutation. Basic facts of DNA replication. Complementary base pairing. Complementary base pairing rules state that cytosine always pairs with guanine and adenine always pairs with thymine in DNA. So the complementary base pairing rule is simply cytosine pairs with guanine and adenine pairs with thymine. Purines, small word, big base. If we compare these two words, purine and pyrimidine, purine is a small word. However, they are big bases. Adenine is a big base. It is a double ring structure. Guanine is a big base. It is also a double ring structure. So purines are adenine and guanine. They are double ring structures. Pyrimidines with the Y are cytosine and thymine. Thymine is a single ring structure. Cytosine is a single ring structure. Each base pair contains one of the other. Adenine and thymine go together. Adenine has a double ring. Thymine has a single ring. Guanine and cytosine go together. Guanine is a double ring structure. Cytosine is a single ring structure. Chargaff's rules. What do you think these lines in between these base pairs represent? If you said bonds, you were correct. There are two bonds between adenine and thymine, so that means there is a double hydrogen bond between adenine and thymine. There are three bonds between cytosine and guanine, therefore they have a triple hydrogen bond between guanine and cytosine. Remember that the bonds holding these bases together are weak bonds, weak hydrogen bonds. Process of DNA replication. The first thing that must take place is helicase, which is an enzyme, is going to come in and break those hydrogen bonds in between those bases, enabling the DNA molecule to open up. So the first thing that happens, make sure you notate this word, helicase, comes in and breaks the weak hydrogen bonds between the bases. The next thing that happens is DNA polymerase, which is another enzyme. Remember, enzymes end in ASC, will come in and start adding complementary bases to both sides of this original DNA molecule, resulting in two separate DNA molecules. DNA polymerases are enzymes that bring in new complementary base pairs to build the new DNA molecule. So remember, helicase comes in and breaks the hydrogen bonds between the bases, and DNA polymerase comes in and starts adding those new bases to make the new DNA molecule. The replication fork is an area on DNA where helicase is unzipping the DNA molecule and DNA polymerase is coming in and adding those new bases. One thing you want to remember is that DNA molecule, is a very long polymer, and you can have multiple helicases breaking up that DNA molecule at different areas of the DNA strand, and you can have several DNA polymerases on different areas of the DNA molecule coming in and adding those nitrogenous bases until the whole DNA strand has been replicated. Semi-conservative replication. Big word, simple concept. Semi-conservative replication simply states that each new DNA molecule contains one old strand and one new strand. This represents the old strand, and we can see here DNA polymerase has come in and is now adding new complementary base pairs. This will represent the new strand. This is just a little video clip showing you how these base pairs are coming in and creating these new DNA molecules. Alright, this is just a quick chart showing some differences between DNA and RNA. The sugar found in DNA is called deoxyribose. DNA is a double-stranded molecule. Thymine bonds with adenine in DNA. In RNA, the sugar is called ribose. RNA is a single-stranded molecule, and uracil bonds with adenine in RNA. Remember, there is no thymine located in RNA. Okay, so we're pretty much finished. This is just another picture of DNA and RNA. I would like you to be able to tell me how they are different and how they are similar. If you have any questions, please send me a message on Remind or write your questions down so that when you return to class, we can have a discussion about what you've learned. Thank you and have a good night.